So hi everyone, I'm Alice Malivert. I'm a biologist. I'm actually a plant biologist, and I started learning AI stuff very recently, almost a year ago. And so this is part of my journey through how to use AI to help me with my research. So during my PhD, I did lots of microscopy, and then I focused a bit more at the larger scale, like looking at the plant, the whole plant, at the end of my PhD, and that's also what I'm doing now. And so I got very interested in using AI for image analysis. And actually, that's a booming field in biology right now. Oh, I have that. Okay, so briefly the outline. I will start with image analysis in biology, with some strategies that people can use, with the model libraries that are being used by the community, ending with a cool new tool that we have, the segment anything model. And then I will focus on agriculture, because that's my particular interest. And with AI image analysis for agriculture, like AI in general in agriculture, then image analysis for agriculture, and then my current postdoc project. And finally, we will talk about some additional material that could interest you for your posters. But basically, AI in image analysis in biology is huge right now. I just went, well, a few months ago, I went to a conference about microscopy. Half the talks were about using AI for image analysis, and the other half was using AI to make better microscopes, basically. So yeah, as you can see, the future of bioimage analysis is a huge topic these days. This is the issue from Nature Method from July 2023. And okay, I did not read it fully, but it talks about AI. So first, image analysis in biology. So there's been a huge culture change in biology in the recent years. So that's from a paper from 1972. So we have some microscopy. Here people are looking at microtubules. Microtubules are some protein polymers basically in your cells that can support a cell, for example. And so they make these long structures, those polymers here. And so people were looking at microtubules a long time ago already, but the kind of microscopy they were doing was very qualitative. So here they show these two images, and then they say, we have polymerization, we don't have polymerization. That's or slight polymerization, whatever that means. So things have changed a lot right now. So, for example, that's what I did with microtubules. Oh, I didn't change the date, but that's not 1972, that's 2021. That's what I did with microtubules during my PhD. So I looked, before he was looking at them in vitro, I was looking at them in vivo, so in the plant. And so this weird puzzle shape are plant cells, and the white filaments we can see here are microtubules. And so now when we look at microtubules, we don't say there's polymerization or no polymerization. We quantify everything we can. So here I quantified the anisotropy of microtubules, so how they were distributed in the cell, if they were all like parallel together, following the same direction, or if they were in all directions at once. After doing a mechanical test called an ablation, for example, usually we change the anisotropy. It becomes higher. They all go to the same direction. And we can also quantify, for example, the angle around the ablation. So basically, now when we do microscopy, we want to quantify everything we can on the images. Not just say, oh, that's a representative image. Look at it, it's pretty. And so to quantify everything, we need to have good tools. The tools that we use are informatics, and so bioimage informatics is a new field that developed with the advance of computers and also the democratization of computer skills to researchers and in particular biologists. So you needed biologists who were able to code and to use the tools that we have. And so, where was it? Some machine learning was, I think there was something for, deep learning democratization was in 2018 in biology, so that's still very, very recent. So everything that I'm talking about, it's like exactly what's happening these days. So also there's been a real boom in the quantity of images. So like these are examples from my PhD, some of the data that I have. I have all of these images that are cotyledon, so like the first leaf. And I have 3 biological replicates, 4 technical replicates, 4 conditions, etc. I have here another kind of image that I'm taking. So for the first one, I wanted to take the size of the leaf. And so what an old school biologist would have done would be to like just click around the whole leaf and get the area from that. Now we are looking for ways to automatize that. Also another kind of images, I wanted the area of white cells, or here we want features describing these cells, or we want the length of that. There's plenty of different things we want to extract from images, and we have lots of images. I know when you do machine learning, these do not sound like a lot of images, but actually it's extremely time-consuming to do it all by hand. And you also need to be trained to do all of this by hand. And so that's, again, time-consuming, and you need the proper people to stay in the same lab, and they cannot move ever because you need them. You also, so you can do it by hand, as I was saying, like click, for example, here, to get the length of that. Or there are some automated methods that are working more or less, depending on which one. So, for example, cell segmentation is not the worst there is. That is the watershed option. So watershed means that you have an image with some white signal here, very bright signal, and some black signal. And do you know what a watershed is in geography? No. Okay. So apparently, I've just learned that a watershed is when you have something in the landscape that pushes the water somewhere. So, for example, you have a mountain that will make that all the water that was on top of the mountain will go to the river. So basically, the same concept. It will find all the high spots, all the very bright spots, the white spots here, and make lines between them. And that are the watershed lines. And that's how you can get segmentation. So I wanted to segment all of these cells individually. And one of the techniques that I've been using with watershed segmentation gives us pretty good results. That's decent. Except that, for example, that point here, that's very white, corresponds to here. And it is not recognized properly by the watershed segmentation. Here we're supposed to have 2 little cells here, and here we have something that belongs, like that they put in another cell, so we cannot have that cell area, for example, and I cannot get these 2 little cells. So it really depends on the quality of your image, of course, and quite often we cannot get the best quality of image possible because of biological reasons. You cannot use all the markers you want in an image, for example. Here I was using microtubules that are on the border of the cell to say that's the border of my cell. But since I did that, there are lots of microtubules everywhere else. Okay, so in your opinion, these blue question marks are questions for the audience. So in your opinion, what can AI bring to the table when we do image analysis? You can, oh wait, yeah, it's on.
I guess it can automate a lot of this and speed up or save a lot of time.
Yeah, definitely. It saves a lot of time. And so
labeling, like you pass an image and an image can, the agent can learn to say this is a microtubule, this was a human having.
Yeah, it can do a lot of, classification is done quite well in biological images. Do you have any
accuracy?
More accuracy, definitely, because when I do it by hand, sometimes I'm not the most accurate person. And also something else, if I do it by hand and someone else do it by hand, do you think we'll have the same results?
I guess not.
Yeah, that's quite subjective. So it also increases reproducibility. What else do we have? Okay, so reproducibility, we said less time efficient, less manpower too, and that is money, manpower. So especially in biology where you want to put all your money into experiments and not into people. We said something else. Less human error. You can optimize the way you will get your analysis. You can use large data sets, something that you would take a lot of people and you would never do it. You can integrate it in a workflow, have all of the different procedures that you want to have. For example, the images that I showed you, they were projection of images because we're taking images in the vertical direction. It's one image, one image, one image every 5 microns, for example. So I need first to do a projection and then to do cell segmentation. So that's a big workflow. You don't require training of people. And you can also do real-time analysis. And that's huge in microscopy because, for example, you can have a microscope that will recognize a cell and follow that cell over time when the cell moves. Or I've seen recently, they even were able to do microscopy on moving, I think it was zebrafish larvae. So like the fishes were swimming in the tank, and there was microscopy, like very precise microscopy, following the fish when it was moving in the tank, which gives you like incredible results, the most physiological results you can imagine. Okay, I talked a little bit about different tasks, like segmentation, stuff like that. What other tasks can AI accomplish? So you said earlier classification, so that was one. Do you have other tasks in mind?
Generating more data.
Yes, that's true. That's debatable. We're going to debate about that right after, but yes, definitely.
Prediction?
Yes, prediction. So that's a booming field. Well, that's a field that we hope will boom. We don't have the tools yet, but yeah, prediction is supposed to help us.
Regression, quantification.
Quantification, yeah, a lot of quantification. So I have it, I think, feature quantification. So I talked about segmentation earlier. We talked about classification. Yeah, I put it here. Data annotation, that goes quite well with classification. Image alignment, for example, you have a movie and your sample is moving. You can get the sample at the same place over time. Tracking, pretty much the same, except that your microscope will move instead of just you putting the image back in place. 3D reconstruction, that's big too. Now we even have VR headsets where you can like see your sample in 3D and like move and like pick the cells, segment the cells. It's becoming crazy. That's super cool. And image enhancement. So yeah, and that's, or even like generation of new image. That's something we should talk about definitely. Because basically you have a lot of hallucinations right now. When you try to do image enhancement. So on one hand, you can get better stuff. On the other hand, is it really the biological thing that you are trying to measure? It really depends. And so these tools should be tested heavily. And right now, I don't know any tool that does not hallucinate something when you try to do anything. So classification, one example here, you have different stainings. I don't know if you can see the image really well. That's the issue with microscopy. But for example, in blue, you have the nucleus. In red, you have the microtubules. In yellow, you have another part of the cell called the ER. And in green, you have a protein, your protein of interest, and you want to know where that protein is localized in the cell. And so this classification says, oh, some of it is in this part of the nucleus, some of it is on the plasma membrane, some of it is no. And then you will know where the protein is. Another example, segmentation, as I was showing you earlier, even with bad signal here, you can get pretty accurate segmentation, so that's really cool. If you want to calculate the area of the cell or the shape of the cell or anything. And image enhancement that we were talking about earlier. Here, I think these are microtubules, and you get the image is way better objectively, and it's gonna be easier to calculate stuff on it. But yeah, some people in my lab try to use these tools and then try to make fake images and see if the AI was gonna do stuff on it. And yeah, it did hallucinate new stuff. So to be taken with a grain of salt. Okay, and yeah, we talked about signal prediction, and so that's something that people are trying to develop. Actually, there's going to be a challenge made by the biggest foundation around microscopy in Europe, coming like next year, on how to do signal prediction, trying to do signal prediction. So if you have, for example, that image of the nucleus, will they be able to find out where the microtubules would be supposed to be, where the ER is, where the protein of interest is? So that's something that could help a lot, but for now we don't have it yet. Okay, and so I was telling you earlier that it's a high number of images. I cannot do everything by hand. It's a big issue. But actually, for machine learning, it's a low number of images. So I'm reusing the same slide to say completely the opposite thing. And so my problem is, how can I use AI if I have such a low number of images? So the strategies that we can use. So in general, to analyze images, we use neural networks. I'm going to explain it a little bit because we're supposed to talk about what we didn't know before we started. And so I didn't know what a neural network was. So you take your data, that here are going to be the RGB values of a pixel, for example, and then you put them into one equation. And then you take the result of that equation and of another, and you create a second one. And all of those are called neurons. And you can have several, all of those are called layers, and you can have several neurons in a layer. And you're going to add everything together until you reach a final number. And that number, you're going to compare it to what the actual value was supposed to be, the prediction. And then you're going to, to what the true value is. So you take your prediction, your true value, and then you put it into a loss function to see how far you were from the reality. And then this comes back to change the weights, so the equation that you had, and to change your whole neural network. And so you do that until you get the right predictions for your true values. And so to train a model from scratch requires a lot of data. And as we saw earlier, I don't have that much data. For example, here that I just went for the vision models, and the most recent one used like billions of images. And I don't have billions of images. I barely have, if I have 100, I'm lucky, basically. So how can I do that? How can I use machine learning to help me with my images? One technique is transfer learning. So I was super relieved when I found out this existed. So you don't have to train your neural network all over again. So here I'm taking the example of a convolutional neural network because they are the most used to treat images, but Rema will talk a bit more about them on Friday. I'm not going to focus on convolutional neural networks here. But basically, you take your neural network, and then it has been trained by someone who has a lot of money, for example, Google, and who has a lot of images. And then you take just the big architecture and all the parameters they have trained, and you retrain it with just your few images. So here they trained it. It was tons of different images, and they retrained the last few layers with someone that was assembling some part of something, like, okay, an assembly line, basically. And then you get your target labels, and that helps decrease the number of images that you need to train the whole thing. Another thing you can do is data augmentation. So that's a picture I took of my plant, and I want to be able then to do transfer learning with my plant pictures, but I still have like 100 plant pictures. But what I can do is change the picture. This will create more data, but it will also make my model more resilient to some changes. So I can do simple things. I just have to think really well about what transformations I want to do. So here I turned it 90 degrees on the right, 90 degrees on the left, a complete 180 degrees. I did some transversal changes. I changed the luminosity, the brightness and contrast. But for example, I don't want to change the colors too much here because I'm studying drought. And so my plants will change colors when it will be drying. So if I put a yellow plant, the algorithm will think that it is drought. So that's bad. So you just have to think really well about it, but then you can do data augmentation and get more data. Another issue is how to find and choose the right model, because we saw there are tons of different models to use. So this comic is showing you how we have lots of competing standards, and then we can think about it really well. So, we have 14 competing standards, that's ridiculous, we will develop our own. And that works for standards, but that works for models. And my model will cover everyone else's user cases. And then we have 15 models. So how to choose the model adapted to your stuff? The solution that biologists have found is a model library. I don't know if it exists in other fields. I hope so. But we have something called the Bioimage Model Zoo. That's a really cool tool. It's a model library for biological images. And so you have several different softwares on which you can use those different models. So you can choose, for example, which software you want to use your model on. For example, I use a lot Fiji. And then you can choose, for example, here you can put a keyword like segmentation. You can put your species. For example, I can input Arabidopsis thaliana, which is a species I'm working on, and they will show me all the models that have been developed with Arabidopsis thaliana. And you will have your model, some examples of data to train it, and how to use it, basically. So that's a cool tool. And it's been developed by a community called AI for Life. So that made me laugh, Nassim, when you presented AI for, I don't remember, for science. Yeah, that's AI for Life. We also have our own AI for something. That's a community designed to bridge life science community and computer science community. So they organize challenges. For example, the challenge I was telling you about earlier, the one to do prediction, they're the one organizing it. But they also try to match biologists with data scientists that can help them. They do pretty cool stuff, and they follow the FAIR principles. So do you know what the FAIR principles are?
Fairness, accountability, reproducibility.
Yeah.
Interpretability, maybe.
Interoperable. But yeah, that's a complicated word. Yeah, okay, exactly. So the FAIR principles have been described to help make machine learning more transferable, and have less redundancy in general. And so yeah, they follow the FAIR principles, and that's why the library of model is super useful. But you still have an issue with biologists in general. For a lot of old school biologists, when you talk to them about code or coding, that's what they imagine. So that's the genetic code. So these are RNA bases, and these are proteins. And so that's not the code you want them to do. But for that, some people have found a way to help. They created an AI analysis tool with no coding skills involved, so that everyone can analyze their own images. So there's a big, that's something we have a lot in biology. You will most often not ask someone else to analyze your data. You're supposed to know how to do experiments, how to analyze your data, and then how to write a paper, and you're supposed to have all of these different skills. And so to have computing skills is not always easy for a lot of biologists. So yeah, that's a cool tool to help them. And so basically it works with the Bioimage Model Zoo that I showed you before, the library. And so you can go on their website, and it's super easy to use. They have basically a notebook for each model, and they tell you, here put the folder where you have your data, and then run it, and then here run that cell, and then here you will have the output folder, run it, and that's it pretty much. So yeah, that's pretty cool. And you can try models with like just, yeah, with your data without training it or with training it with your own data. So that's pretty cool. And that's in the posters that I'm considering for you guys. If you want to do a poster on this tool and the library before, that can be a fun idea. Okay, another issue that we can run into in biology is that we all have lots of different experiments. So here it was a matter of image enhancement, as we saw with microtubules. Here it was a matter of image segmentation, but it's like these were microtubules in vitro. This is a plant in vivo. These are someone wants to segment nucleus of these animal cells. Here you have another kind of microscopy, transmission electronic microscopy, where they wanted to segment these little different parts, the mitochondria. Or here they wanted to do some classification from animal cells. So you have lots of different models in biology and lots of different tasks. How can you avoid training a model for each experiment? And the answer lies in foundation models for me. And I'm going to talk about zero-shot learning model, the segment anything model that has worked super well for my data. Yeah, okay. So during my PhD, I wanted to know, I had leaves of a certain mutant, and I wanted to know where they would fit on a mechanical stiffness scale. So here we have something very soft, here we have something very stiff. Were my leaves soft or stiff? Because I was studying water, and water plays a mechanical role in plants. So if you take a plant and you put it in, so that's onion cells that are stained, you put them in a solution with a high solute concentration, water will go out of the cells. So you see that the cells become completely deflated. If you put the cells back in pure water, the cells will be completely inflated, and that makes them very rigid. And you can see the plant cell as a mechanical system in general. So, for example, you have the osmotic pressure inside, so how much solutes you have in your cell. That changes how much water gets in your cell. And since you have water in your cell pushing against what we call the cell wall, so a layer surrounding every plant cell that's quite stiff, you get a pressure because the water will inflate the cell like a balloon. It will inflate the cell, and that will put the cell wall in tension. And so you can characterize how stiff the cells are. That's very interesting for us because, for example, we want to know if that can change the way the material behaves. The wood, for example, will behave differently depending on the stiffness of the cell wall. That will also change how cells grow. For example, a stiff part will grow less than a soft part. And you can characterize how the cell wall behaves mechanically. Yes?
So when there's more water inside the cell, the cell wall is more tense?
Exactly, yeah.
And the leaf it's stiffer?
So, yes, a little bit because it's in tension, but also because the issue with biology is when you have something, you have another force that will something else that will sense it and react to it. And if you have more tension, usually you will have stiffer cell walls. So first, you're stiffer because you have more pressure, but also because the plant doesn't want the cells to burst. They will create stiffer cell walls that will prevent the cell from bursting, but also be stiffer.
Okay, but at a super high level, when the leaf is wet, it becomes less stiff, right? No? Or when it's dry, it becomes
softer. Okay, when it's super dry, when there's no more water at all, yes, it becomes less stiff. But for example, if your plants go under drought, they do wilting. And wilting makes the leaf softer. Yeah. And so I was wondering all of this because I had a mutant. So a mutant is just a way for us to say that we broke a gene and we were trying to see what that gene did. Because if you break it and you see what's not working anymore, you will know what the gene was useful for. And so I had a mutant called Feronia here that had cells here that burst. They were like inflating so much that they burst completely. And so there was an issue somewhere either in the stress sensing or in the feedback mechanism, like reinforcing the cell wall. We were unsure. And here you can see even like the creases made by those cells that burst, just like subbubbles. These are not cells, these are just subbubble images to make you realize it bursts like a subbubble. Okay.
Can I just ask a question just on the technique?
Yeah.
How do you kill a gene?
Yeah. Okay. So originally it was done randomly by luck. So for example, you expose seeds to UV light that create mutations, or you expose them to chemicals that create mutations, and then you characterize what the seed will give you, and that's a new mutant. Now we can target mutations with CRISPR-Cas9. I don't know if you've heard about it.
Yeah, CRISPR, yeah.
Yeah, okay. So it's a way where, like, you put a piece of, well, it's an RNA, but it will bind to a piece of DNA, and you will say you will do an excision here. You will break that gene.
Okay, cool.
So yeah, that's a really cool new technology that's gonna help a lot in biology, definitely. And so I was looking at my cells, and I saw that those cells swelled until they were bursting completely. So here you see the bursting, it changes the coloration of the cell, and it deflates the cell. They behave like subbubbles. And I wanted to know what the cell mechanical properties were to understand where the issue was. If it was with the cell wall that was not rigid enough, or if it was with too much water intake that made the cells swell and burst. And as we saw earlier, when you have a high solute concentration in the medium, water will go out of the cell and the cell will deflate. And the way cells deflate depends on the mechanical properties of the cell. The way cells deflate depends on the mechanical properties of the cell. For example, if you have a stiffer cell wall, they will not be able to deflate as much. If you have more water flows, the cells will decrease in volume more rapidly, faster. And so to do that, to analyze that, I needed to measure the way cells were contracting. And so I did not do it at the scale of the cell, I did it at the scale of the whole leaf. It was simpler for me. And so I just took a leaf, put it in a solution with a high concentration of solute, and just looked at how the leaf shrank over time, over more than 20 minutes. And so I needed to analyze my images quite fast because I have lots of replicates and I don't want to do it by hand. In the beginning I did it by hand, so that's how an old school biologist would do it. You take a software called ImageJ or FitJ, and then you do 1 point at a time, and it's quite long, and you have lots of images. And it's also not super reliable. So I started looking for ways to segment that. And maybe it's because my images do not have a very high contrast, maybe it's a weird shape for the usual segmentation tools, but I did not find a segmentation tool that was useful for me. I did not try all of them, but I tried a few. Yes, sorry, I should have put that. Yes?
How did you try to segment anything model that you mentioned earlier or not?
Yes. No, perfect. That's a great transition because I don't know why that slide is here and I want to pass it. Okay. So here is the segment anything model, the way I wanted to try to see if the segmentation was possible with my cell and automatic segmentation. So that's a model, that's been developed by Meta. And that can segment literally anything. You give it an image, it will segment it. You don't have to train it. That's what's amazing about it. It's a zero-shot model. It's been trained on billions of images. You don't need to do anything. You just give it an image. So here, a little corgi, and it will segment like the corgi, the trees, anything. You can give it different prompts to say, I want to segment this thing in particular. So here, I want to segment the snail shell. And so I put this little dot here, and it was a point prompt. Here I tried with a little box around the shell, and it gave me just the shell of the snail. You can ask to segment everything in the image. And so I got my shell, but I got also everything else. So that is not accessible yet, as far as I know, but they have developed a way to do it with a text prompt. So say a wheel, or here I could have tried to do it with the snail shell, and then it will segment that stuff. So that's a super cool model that's gonna help a lot, I think, in biology. And that's quite easy to use. Everything is on GitHub and quite well explained. So again, cool, thank you. And so the segment anything model is three things: a task, a model, and a data set. So the task is you have a segmentation prompt, you have an image, you put them into the model, and you get a mask of what you wanted to segment. The model, I'm going to go back on it a little bit, but it uses a transformer. And the data, I'm going to talk about it right now because I only have that slide. They use more than 1 billion masks, which is like a huge number of masks that they could use. 11 million images, all that were supposed to be privacy respecting, and that were supposed to be like super fair and having images from all over the world with like an equal representation of each country, which did not happen. An equal representation, okay, there is an equal representation of each gender, but there is not an equal representation of each age group. But yeah, okay, lots of images. And what's been super cool also is that they used, so they input some data in the model in the beginning. Can we see my, yeah. They trained the model with some data, and then they use the model to annotate more data. And they use that data to train the model. And then they use that, I think they went 3 or 4 times over it. And so that's the way they were able to have more than 1 billion masks without too many people on it. Okay, about the model. So the model uses a transformer. So you have an image, and you have an encoder making an image embedding, and then you have a mask decoder that takes into as inputs the image embedding and different prompts. So you have, so the encoder is a pre-trained vision transformer. So that was actually even pre-trained to help them a bit. The prompt encoders, you had positional encoding for point and boxes, and for text you had clip, which is basically the thing they use for DALI, if I remember correctly. And then you have different masks produced with different scores of how accurate they think the mask is. I'm going to talk a little bit about transformers because that's one of the ways that you can analyze images. So there are basically 2 ways to analyze images right now that are competing: CNNs and transformers. And so I'm not going to talk about CNNs. Rena will do it on Friday. But I learned a lot about transformers. So what you do is, just as a normal neural network, you take your input. For transformers, your image is, you divide your image into little parts of the image, and you treat them the same. And you have also a positional encoding saying that part was there in the image. And what's different with a normal neural network is that you have attention layers. You have self-attention in the encoder. So a transformer is an encoder or several encoders and a decoder. And so you have self-attention layers in an encoder and encoder-decoder attention plus self-attention layers in the decoder. So you make your data go through all of these layers and then go to the decoder, and this will give you your results. And so how an self-attention mechanism works is that, for example, if you have a sentence, it will look at all the words in the sentence and try to relate each word with another word in the sentence, if it can. For example, here we want to know if she is related to doctor, nurse, question, whatever. It will go through all of the text and see that it's more related to nurse than to something else. And for he, it will go through the text and see that it's related to, well, more to he. It didn't work well with the doctor here, but yeah, you get the gist of it. And why is that super cool? It's because then the model gets an idea of the bigger picture. It does not take just a little part of an image into account, but like the whole image into account. So that makes, that makes it easier to see a whole picture and to see important stuff happening at a large scale. It's okay, and another thing I was telling you about the encoder-decoder attention, it's to see if the input and the output have similarities. So, for example, here you have a sentence that's translated from English to French, and the encoder-decoder attention will see that there are more similarities between the words in the middle, except for those three words, because in French you invert the way you put words sometimes compared to English. So, for example, European will be the last word, whereas European would be the first word that you put. And so that's what makes transformers powerful. They can first see the bigger context. Second thing that I forgot to say, they can parallelize a lot of things. So if you have several GPUs, for example, you can run everything on different GPUs and get something very fast. And they also can take into account the input and the output a little bit, which helps make something more coherent. Okay, so what happens with my images? What I did was that I imported the SAM functions, defined a point prompt for all of my images. Because they're not moving a lot, it was really easy to do the point prompt, the easiest for me. And then I calculated the different masks, and I chose the best one every time. And then I did that on all of the data and returned a list of my mask areas, because I was interested in the area of the leaf. So if I do that, I have the three best masks according to the segment anything model. The one I want to choose is the second one, because it takes the little stem here. But yeah, every time I can choose the one that will be the best. I actually create all of the masks and just choose the one that looks best. Overall, I had impressive results with all of my leaves. I was very happy with it. I had some issues though. For example, if there's a bubble that went on the edge of my leaf, it was a little bit harder for me to get it. And sometimes, due to contrast issues, I think I could not get the whole leaf. So ways I found to navigate around that, I have to try. I haven't not done it yet, but the box and the text prompts, because yeah, maybe here if I put that part into the box, it will be a bit easier. I could try to extract the mask and then manually change the mask, but that kind of defeats the purpose. I could try to alter the images. I think that's one of the easiest things to do here. For example, I could put a black line here over the bubble end, and it will just take it, except that will not be the most accurate way to measure it. Or I can just collect more data, because collecting data is super easy with that experiment. And if I can just batch it, like run it through the segment anything model, well, it's the easiest thing for me. So that's the thing I'm doing these days. And so first, some preliminary results. So in red, you have my data. In white, you have the model that I'm trying to fit to the data to see how mechanical properties will change along time. So that's a pretty good fit for biology, even if it does not look like perfect. Some very interesting thing that I found is that my mutant, the Feronia mutant, have some really weird behavior compared to the control. But that's actually very probably because cells are bursting over time. So that gave me the idea to look for cells bursting during that plasmolysis. So that's something I'm going to do later. And yeah, I already found some, so this is really preliminary data, but I already found some different characteristics between the mutant and my control. So, for example, here we have a different elastic modulus, a different rigidity of the cell wall. And so we think that my mutant is more rigid. And you think that's a bit counterintuitive because you think it's more rigid. Why does it burst then if it's rigid? But actually, if it's more rigid, it's less elastic. And so it can take less pressure into account. And so it will break before deforming. And it also has a high hydraulic conductivity, which means that there's a faster water entry. And so there will be more water getting in, and it will not be able to deform, so it will break, basically. That's just preliminary data, but that's a pretty good sign for us. Now I'm collecting more data. Okay. Oh, I'm very fast. Do you want a short break? We have 5 minutes. Yeah? Let's have a short break.
Can I ask a dumb question on
Oh, yes, sure.
the slide before?
Yes.
I see these plots a lot. I'm not used to them in physics. How do I interpret what all the different things mean?
Yes. Okay, so this is our box plots.
Yeah.
What you have is here either the mean or the median, depending on who is doing it, and they should be saying what it is, and I did not.
Yeah.
Here, I think it's the mean for me.
Where am I looking? Okay, yeah, the central
The central bar.
Yeah.
Yeah, sorry. Oh yeah, can you see my laser pointer? Yeah, a little bit, but it's a bit hard.
No, we can see.
Okay, and where's my... Yeah, and here the outer bars are standard deviations, usually.
Okay, and then the coloured box.
So each box is a different sample. So here you have my control on one type of growth medium. Here you have my mutant on the same kind.
So it's like these outer. What was the standard deviation?
So here you have, yeah, I forgot to say, sorry, first quartile, third quartile,
Yeah.
and I think that's standard deviation.
Okay, right, yeah, okay. And the black, sorry, I just tried to understand.
And the black dots are the data points.
That's the data.
So I have a very low number of data here. It's very preliminary data.
Yeah.
There was basically, like, I shouldn't have done stats on them, but that's fine.
That's fair enough, yeah.
But yeah, so I can talk a little bit about data visualization because I like it. So basically, a lot of biologists will use bar plots, histograms, to show their data, which is bad, and you should not do that because it hides a lot of the data. I like to show data points in general to see what's happening. And if I have too many data points, I will not use a bar plot. So the bar plot will show a bit, like a few statistics on it, but there are other ways to do it. I like to use violin plots.
I think you have one. These ones that have like the nice...
Yeah, the violin shape. Yeah, I had one in the beginning, yeah. We're gonna go far. Yeah, sorry, I'll always think that it's super obvious, but no, it's not. Even for biologists, like violin plots are not super used. So if you have too many data points, you don't want to show them all. What you can do is show the distribution on the side. And here you have, I think that's the mean and the... Is that the mean or the median? No, I think that's the mean. Like first quartile, third quartile, somewhere here. Yeah.
Yeah, nice. There's a lot of information.
Yes, exactly. You don't hide anything. For example, here we have something that's not exactly Gaussian, but that's the best you can do for Gaussian in biology. But here you may have something that a little bit has, how do you call it when it has two different...
Multimodal?
Yeah, multimodal. Maybe, a little bit. But yeah, it's not strong enough that I can say anything.
Cool.
Yeah. Okay, so now the 5 minutes break. But if you have any question, please ask them. Okay, now I'm gonna talk about the case of agriculture. Because I work in plant science, and every time you want to sell something or try to figure out why you're useful to society, you use agriculture. So, uses of AI for agriculture. First, well, agriculture is essential to like feed us and like dress us and stuff like that. And it's a sector that will face new and greater challenges with climate change in the coming years. And so, for example, global warming plants do like a certain range of temperatures. So yeah, I can see it in my growth chamber when Imperial has issues with heat. It's an issue, definitely. You can have extreme weathers, you can have difference in rainfall intensity. The soils have been depleted a lot also during the past, since the Industrial Revolution pretty much. The agronomical revolution more, but yeah, it's quite close. So basically we have tons of issues and we're still trying to feed everyone and the population is growing. So we need to revolutionize agriculture pretty much in the coming years. So there are tons of ways to do that. But machine learning is predicted to have a huge impact on the market. So that's the 27%, the part that's supposed to that machine learning is supposed to bring to agriculture in terms of growth in the years to come. And for example, in agriculture, they love Internet of Things, so that's not exactly machine learning. But for example, like when you have a smart fridge or smart, in that case, it would be, how do you call the place where they store all the things they need for agriculture? Like, for example, all the nutrients that they will, all the fertilizer, like un entrepôt. A warehouse, thank you, yes. Okay. So you can have, instead of a connected fridge, you have a connected warehouse. That's the idea. And it's supposed to boom in terms of market values to $4.5 million. So smart agriculture is a lot of talk right now. Lots of companies are super interested in it. I'm going to talk a lot about Microsoft because I've been to a conference where Microsoft was talking a lot and they're super involved in that, but I'm sure lots of other companies and very probably smaller companies, more representative of the world, are also doing. Yes?
Just to go back on that $4.5 million.
Yes.
What's that represent? Because that's kind of small, right? If we're talking about, what is that the cost of one warehouse? Is that what you're saying?
No, for me it's like just like how much selling stuff with Internet of Things for agricultural purpose.
Okay, so, yeah. Sorry.
Yeah, no, it's fine. So, first question, how can AI be useful for agriculture, in your opinion?
Less manpower, right? You have monitoring systems doing that for you.
Yeah, monitoring, definitely. That's the major part, I think, of it.
More productivity, like yield.
Yeah, you know what your yield is going to be, definitely. Yeah, so you can, for example, plant the right thing because you will have a greater yield.
Yeah, I suppose you can also do soil analysis, so you're not over-fertilizing or under-fertilizing. Or
yeah, exactly. You can do, monitoring and, also analysis of your things.
Filtering out what you harvest, like faulty, vegetables, like a computer vision system that can distinguish between things that are you throw away and
yeah, that's cool. That's something I did not consider when writing that, but yes, that's definitely already in process, I think. Like, I know that there's this company that made like a tool that will recognize different kind of baked goods, like over hundreds of baked goods, so that then the cashier, like in a bakery, will take all the baked goods and it will calculate the price of the whole thing, so that people will not have to know by heart what hundreds of baked goods are. And it's also more sanitary because they don't have to examine it. So I guess that works with like apples, you know. What do I have? We talked about predictive yield. We talked about soil monitoring. In general monitoring, you can also monitor crop health, like know if you have a pathogen or something in your crops. You can have genetic optimization. You can use machine learning for genetics in general and know which crop will be the best to put at that place, for example. You can have fast answers to questions like, I'm in my field and my cow is looking sick. She's, I don't know, What can a cow do? She's pooping weird. What's happening? Well, ChatGPT can answer that. You can have market prediction to know what to sow, disease diagnosis. You can have real-time weather, like monitoring of weather. You can have micro-weather predictions, so that's big, apparently, in agriculture, because when you look at the weather, you want to know if it will rain in 1 hour. Because, for example, you cannot put fertilizer, or no, it was pesticides right before it rains. It's bad for the plants. Or it goes away, I don't remember. But anyway, you want to know the weather exactly in your field. You can have supply chain optimization, so that's the Internet of Things. For example, you have a connected warehouse that will order for you more fertilizer when you're done with your fertilizer. Okay, what kind of models can be useful? I found a few examples with regression, clustering, Bayesian models, and neural networks. Do you want to give me ideas of what kind of tasks they could do with tasks we saw earlier? So, for example, what could you use clustering for?
With respect to what was on the
Yeah,
previous
exactly.
slide?
Yeah, or new ideas if you have any new ideas.
Similar genomes or something.
For clustering, yeah, okay, yes, definitely. You can have several, you'll put all of your crop varieties into different weather conditions, for example. That works. Do you have any idea with Bayesian models? Yeah, for predictions, exactly. Yeah, like how much, yield you're gonna get from the conditions that you have. So let's see a few. So regression, you can predict crop yields with fairly simple stuff. You can have the price of input, the price of output that you can predict. Clustering, you can choose the right crops. That's exactly what you said. I had the exact same example to grow depending on the environmental conditions. You can do predictions with Bayesian models depending on what your environment is like. Neural networks are used obviously for image recognition. We talked a lot about it earlier. For example, you can do pathogen detection, and you can analyze complex signals if you have a very complex answer to an environmental condition, for example. But what risks should we keep in mind if we use AI in agriculture, in your opinion? Like, what could go wrong?
I think human error is probably, I don't know, human expertise and machine expertise, the error is probably different that sometimes you might want to rely on a human.
Yeah.
Something like agriculture versus a machine may have high variance or something.
Yeah, we definitely want to keep an eye on what they're giving us, especially now that it's in development. Yeah. Do you have any other ideas?
I guess you need to have like reliable models or something. Like if in your connected warehouse one of the detectors stops working and it just orders the same thing
Over and over. That can create issues. Yeah, definitely. You're gonna have system issues at some point. Yeah. Okay. These are a few ideas. System failures. That was what you were talking about. Other issues that you can have, you have to decide what to prioritize in your model. Do you prioritize yield or do you prioritize environmental consequences? And who's making this decision? That's a big social question. Like, for example, will I put lots of fertilizer on my soil? And if I prioritize yield this year, do I want also to prioritize yield next year? Like, you have to think about all of this when you're making a model, and
But how is that a model thing? It's also like a non-model thing in agriculture, right? Like you could also not be using a model and still be prioritizing yield.
Yeah, definitely. But your model is gonna tell you, for example, if you take your model without thinking, you just look at it, tells you, you have to put fertilizer. You didn't make the choice to prioritize yield over environmental consequences. Whereas now you do it without a model, but you make the choice. Or in Europe, the agricultural politique agricole commune, the regulation is doing that choice for you. You know, like who's making that choice? Is it politics? Is it agricultures? Is it companies that are selling you the model? It's a big thing. You can have cyber attacks. We see what cyber attacks do to hospitals. Imagine if you cannot feed your country because there are cyber attacks on your farms. That can be a big issue. System failures, data stewardship. Who does because every time you're putting data into your model, does the data belong to you? Does the data belong to the companies that's selling you the model? What's happening? Can they use the data? Lots of questions to think about, and socioeconomic inequalities. So right now, ML is already in use in some big farms, big scale things, but a lot of countries rely on subsistence farmers, like making very small farms. And these farms most often cannot afford satellite images and ways to analyze them. So you have to think about how they're going to do it if they want to get to the next step. There's a paper on that, a super cool paper on how ML can be useful for small farms. And it's in the poster ideas that I'm offering. Okay, so now AI image analysis for agriculture. So here I'm going at two scales, the large scale, satellite and drone images, and then at the smaller scale, so just walking in the field looking at your plants. What can we use? This is drone images. What can we use drones for in agriculture? A lot of big farms are already doing it, actually. You can do terrain mapping, for example, if you want to know what crops you want to put at some place. You can do equipment inspection, everything remotely. You don't have to get up from your couch and you know if your big machines are going well. You can do livestock monitoring, just follow your cows with a drone instead of sending someone. You can do soil and crop analysis and monitoring, but you have to recognize the images in all of the situations. You can do apparently seed planting. The drones can carry seeds and then just plant everything. You can do crop spraying, so that's the image that I put. And I forgot here, but I know people who are developing a robot. It's not a drone, but it's a robot on wheels to pull out weeds, for smaller farms. So you can do cool stuff, actually, that require less manpower. And with satellite images, so first, satellite images are quite accessible data. You have to pay for them, but then you don't have to have the equipment, so that's cool. And anyway, this data is being taken by satellite all the time, so you're kind of exploiting the data that's already there, so that's also cool. You can measure a vegetation index, like here in this picture, know where your crops are or where the forest is. You can measure stress index. Quite often they can measure, for example, the temperature, and that gives you an idea of the stress the plants are going under. So for example, here there's a high stress here. You can measure where there is water or natural resources. So for example, here we know that there's a high stress, but there's not a lot of water. So if there was a flood, for example, the stress that these plants are under is not caused by the flood. You can do disaster response. I was talking about the flood. You can check what's happening in real time. Well, not exactly real time, but you can check how it has affected your field, for example. You can do yield prediction, planning how you're going to use your land, and weather monitoring. The thing is, with satellite images, you have one issue. You need a post-treatment. So that's something developed by Microsoft called Space Eye in their big Azure, thank you. Cloud thingy that can erase clouds from satellite images. So the way they do that is that they take radar images that gives you an idea of what the material is, and they use old images, satellite images without clouds, and they use both the radar and the old image to create a new image. So this is to be taken with a grain of salt, of course, because the radar see like the material, but it does not see the color, for example. So you cannot use it to say, oh, my crops are super yellow, they need to be watered. But you can see, for example, where there is a lot of water and stuff. Okay, they also developed something else to an image enhancer to get better images from your satellite images and get, better results. And what's interesting is that Microsoft is developing a platform called Azure Data Manager for Agriculture. So for now, it's in preview. They were doing something very similar, and they changed the name, if I understand, from, it was something with farms inside. Well, if you've heard about it, but anyway. And so basically this platform would be for farmers to put their data, and they can analyze their data, their farm data on that platform automatically. They can manage their data, and there would be apps connected to that. For example, you can connect drones directly to the server, and then you can look at the images of drones will be put directly, and you can, for example, order a drone to do something from your app, if I understand correctly. They will include models that can do some data analysis and do predictions and stuff like that. And Bayer is a partner that will help, apparently. So that's an idea of what could happen. So we could have completely connected farms in the future with the help from big companies. And just because I wanted to talk to you about the segment anything model again, so we had those big images of fields from satellites, but how can we exploit them? Well, for example, we can segment them and measure the surface. So you can do it with here, this column are drone images. So here you can see exactly how much area of, I think these are trees, you have, or like bushes. You have it for airborne images, like from planes, or you can have it from satellite images if you want a larger scale. So this can be super useful to detect stuff. Especially, we discussed with Nassim earlier, if the prompt thing, the text prompt thing is usable, you can say, calculate the area of the forest, or tell me how much water we have, stuff like that. So that's going to be very useful. One issue with satellite images and drone images is that it's not always precise. So here we have an app that's supposed to tell you if you have stress in your plants. So I think the green is good and the red is bad, stuff like that. But once you have that, you know that your plants are not going well, but that's all you know. Because, for example, you can have very different kind of diseases. These are two different kind of things that are affecting wheat. Here you have a fungus, here you have a bacteria, and that will require a completely different treatment, but you cannot say that from satellite images. So you need tools at a lower scale, at a smaller scale. So diagnosis and augmented reality. Biologists are really into augmented reality these days. That's a big thing. So we already have some diagnosis apps that are existing. I tried one on my phone. So you point it at your plant, like your house plant, and you say, what's wrong with my plant? And they say, you should water it, or it's got a fungus or something. So most of those apps, you have to pay for them. They can recognize your plants and stuff. That's pretty cool. And so that's one way to do it. Or you could even have a chatbot that includes these images. So with the newer version of GPT, you can include an image prompt. And so you can say, oh, here is my sick cow. What's wrong with my cow? And it will tell you, oh, that's what's wrong with your cow. And then you won't have to go and see a vet. Or you will have to go and see a vet. It will tell you probably to go and see a vet. So yeah, that's what we have for now. But we can push it to the next step with augmented reality. So you very probably know augmented reality from Pokemon Go on your phone, for example. So you can carry around your phone and look at your cows. Or you can even have headsets. So virtual reality headsets are even like a step further. Well, augmented reality, sorry. Headsets are a step further. And so basically right now a lot of companies and like universities are trying to develop augmented reality tools to do diagnosis. So here are examples from the Wageningen University. So they do lots of crop research. And so they're developing, so I think that was an image for a master internship. They're trying to make a master student develop an app to use augmented reality and see what's wrong with the crop. But I know that they are also developing like headsets to see what's wrong with the crop. Like just walking, imagine walking into a growth chamber and like you look around and it tells you that crop, that plant is unwell. You should put pesticides on it or whatever. So that's that's what we call smart farming. You don't spray your whole plant, like all of your plants. You spray just the plants that's unwell, and you get it really fast in real time. So and again the segment anything model, because I really like it. You can use it for augmented reality too. So imagine you put on your headset and then you look at your cows and it will tell you that's what's our typical name for cows in the UK? Marguerite in French, but Daisy? Okay, that's Daisy. Yeah, for example, that's Daisy. Daisy is here. That's good.
Marguerite.
Yeah, Marguerite. Okay, how much time do we have left? Ooh, still a bit early. Do you have any questions?
Wait, why do you even need augmented reality to say the specific cows that you can
To see that all of your cows are there.
I can still see with my eyes.
Oh, yeah, okay. Yeah, but if you have several hundreds of cows, I guess you don't have to learn their names, all of them.
Oh, I see. So each color knows which cow it is.
Exactly, yeah. And if we count the cows and say, for example, and record like in a table, Daisy was here at that time. You know.
So if I understand correctly, it doesn't just segment them; it classifies them.
Yeah, but you need to segment them to classify them.
Yeah,
Yeah, or like if you're ecologist, for example, so that's not agriculture exactly, but you can look at the cow behavior once it's segmented. You can see that it's a happy cow walking. You can say, for example, the green cow is a leader cow. That's a thing with cows. They have leaders like that will tell them, you go there. Yeah, stuff like that. Okay, more questions? No? Okay. So now I'm going to talk about my project. So I'm studying drought stress, so lack of water, and its impact on plants. And I'm trying to develop a platform for early drought stress recognition in plant leaves. So I'm focusing on drought because it's one of the primary causes of crop loss worldwide. It's responsible for 34% of crop losses in low and middle income countries. That's the highest kind of stress you can have, like the stress responsible for the most crop losses in low and middle income countries. And so we want to find management methods and crops with a better resistance to drought. So what I can do to measure drought is, for example, look at the moisture levels in my soil, and I have instruments to do that. That's super easy. But if I want to see what crop is better resistant, like what variety has a better resistance to drought, I will give them the same treatment, so the water in the soil will not help me. I will need something that analyzes the plant response. And so I want to be able to detect drought stress fast, simply, and cost efficiently. There are several,
Is the idea that if there is a drought-resistant crop, then you find the mutant or the gene and then you modify the other plant?
Yeah, I could do that, for example. Or I could say, for example, what I want to work with is the management methods, because there are tons of social issues with crop genetic modification, but that's another topic. The management method I'm working on will probably make crops more resistant to drought. So we roll on plants, like we submit them to mechanical stress by rolling on them or like walking on them, and they should be better resistant to drought, resist better to drought. So yeah. And so to do that, we need to be able to see if the plant is going under drought stress or not. So there are several ways to do that. In the field, you can just look at plants After sometimes you develop an eye for it. I can say if my plants are unwell. Like, it's actually a little bit more of a touch thing than eye thing for me. Like, when I touch them, I know if they're well or not because of the stiffness, as we talked about earlier. And that's a fast and simple way, but it's super subjective, it's qualitative, and it requires training, and it takes you a little while to be able to detect stress. You cannot see it in the first days, whereas the plant is already suffering, you know. In the lab, we have tons of methods. We can look at the aperture of stomata. So stomata are little holes on top of leaves that let CO2 and water and O2 pass through. And so usually they tend to close when they're under drought stress so that water will not evaporate from the plant. They will retain water, except that then you do not have carbon dioxide entering, and so you will not have photosynthesis. So you're not happy when that happens. And so to measure that, you need a microscope and some semi-complicated techniques. Or, for example, you can look at gene expression. So you have to freeze your plants in liquid nitrogen, crush them, extract the RNA, get the RNA from RNA to DNA because it's more stable, and then look at how much DNA you have, and that takes, and of which gene, and that takes a lot of time. It's expensive, but even if it's quantitative and reliable. So these options are not good either. So what we are doing is that we are taking a leaf from the farmer's book, and we just want to look at the plant and to be able to say this plant is under drought stress. Also, this could be combined in an ideal future with like the VR headsets. You look at your plant and it tells you what of that plant. That could be cool. Yes?
So you mentioned that in your case, like, it has a lot to do with the touch.
Yeah.
But do you think there's just a visual signal, but it's just harder to see and touch?
I hope that. Yeah, I'm definitely hoping it. It's also because that's something I'm more sensitive to, the touch, but someone else will be able to look at it and say just by looking at it. Yeah, that's again one issue. It's super subjective looking at the plant. And we want to make it more objective. So I'm trying to build an AI tool to detect drought stress from plant leaf pictures. So what I'm doing is that I take plants, I stop watering them after some time, and every other day I'm taking pictures along with quantitative parameters. So I talked to you about the stomata aperture and the gene expression, but I also measure how much water there is in the leaf. It's called the relative water content, compared to how much water there could be. I measure some photosynthetic parameters, so if the plant is doing photosynthesis well or not. I measure ion leakage, so when cells die, they release ions that were in the cell. And so if usually under drought stress, there will be more cells dying, and so there will be a higher ion leakage. And I can measure the soil moisture. And so I will put all of this, along with the images, into an algorithm that will tell me, if I give it an image later, that plant is under high drought stress, well, that plant is under low drought stress, it's fine. So as a proof of concept, some people have done similar stuff, not with drought, but with other kind of stresses. So for example, here they use soybean leaves and created a model called XPLNet to classify those leaf images into different kind of stresses. So you have healthy, but you have herbicide injury, and you have different ion deficiencies, or well, nutrient deficiencies, different bacterial issues, stuff like that. And so, and then what's super interesting about that thing is that they managed to have an explainable framework. So after the classification, they asked the algorithm, what did you use to do the analysis? So here, I think they used the most activated pixels, like the pixels with the highest activation, I think, in the CNN. Anyway, they were able to say, so this has been classified as herbicide injury because it has spots here, here, here. And so what I want to reproduce more or less the same thing, because it was explainable, and something else, it's transferable to other species. So they've done it with soybean leaves, but it worked with very, very different leaves. So that's very interesting. You only have to train it once. So right now I'm collecting the data. I actually took that data point yesterday, and I'm taking that one tomorrow. And yeah, I'm taking pictures along with all of those different parameters over 22 days with like lots of plants. Well, this is a lot to me because it is taking me 13 hours to take all of these parameters with like 16 plants. 16 times 2 because I have my control and my drought experiment. So, but in the end, I'll have 120 images for drought stress and 120 for control. I will reproduce that maybe once or twice, but I cannot do that experiment forever. So I want something that will use not a lot of data. So my question was, should I use a convolutional network or a transformer in the beginning? We talked a bit about transformers. I'm not going to talk too much about convolutional networks, but the pros and cons. Basically, CNNs have a lot of are good for spatial hierarchies. So if you have spots, for example, at a certain part of the leaf, it will recognize it. So that's good for me. It can do, you can do parameter sharing. There is less data required for transformers, and I do not have a lot of data. It requires a lower computational power. And you should talk to Michael about computational power at IX and how we need our own thing. But in the cons, it's a limited global context. It will not see like the shape of the leaf, which maybe the leaves are more round when they have more water. I don't know. And it's a very task-specific architecture, whereas transformers are really good at understanding a global context. And they're more versatile, but they require lots of data, lots of computational costs. So in the end, I chose CNNs.
Why can't you use like GANs to generate more images? Like you have 120 images, you put
because I don't know how accurate they will be, like how close to the real data they will be. And I'm also using, so if I generate more images, I need to generate all the parameters, like all the physiological parameters, the stomata aperture, and it's a lot of things that we don't know anything about, actually. We don't exactly know how drought works, which is an issue that I'm going to talk about a bit later.
Oh, I see. So when you create an image, you have to generate a bunch of like features.
Yes, exactly.
So if you were to generate image through a GAN, then there's no way to generate that.
Yes, exactly.
I see.
Yeah. And so I chose a strategy that's adapted to my small data set. So first I'll do some data augmentation, and here I will keep the same physiological parameters. So it's new images, but with the same parameters, so it works. And then I will do some transfer learning. That's why I was telling you earlier about the transfer learning. So I found a few models that should be working quite well because they've worked on plants already. For other stuff. And the first model is the one that I was showing you earlier that detects stress and classify things. And then I will try to do drought stress classification and get an explanation for the model. But there's an issue with that. Can you find out which one?
It's specific to the type of plant?
Yes, it's specific to the type of plants. I hope it will be transferable, but I have no idea. I have another issue, it's with the classification. We were talking a little bit about it earlier. Do you know what could be the issue?
Well, is it because it's qualitative?
Yes.
So you need to
decide.
Define what your label is. Yeah.
That's exactly that, yeah. That's my issue with it. How do I say that's low level, that's high level, and that's in the middle? How do I even define how many classes there are? Also, to create my classes, so in the beginning I was thinking, it's easy, I stop watering them at the same time, and then I'm taking every day a certain number of plants, they will go together. All the plants day 4 of drought will go together, that's a class. And I was wrong. I was cruelly wrong because my growth chamber is super variable, and also there's a lot of variability in any biological model that you have. So it's not an accurate measure time after watering. So here I'm showing you, where's the red? Here. This is a relative water content, so how much water there is in the plant compared to how much water there could be. And here the time point after drought. In blue I have my control, which stays pretty much the same. And in orange I have my drought stress plant. And I have this amount of variability at day 18. So that's not a good sign. I should not use the time points. Yeah, especially since there have been more issues with the growth chamber, and it's, really, really bad. So I'm looking for a way to actually, quantify drought, which has never been done, as far as I know. I have not found a model saying, oh, you should just, I don't know, multiply the stomata per the relative water content, and you have a drought index. So I'm defining a drought index. And how will I do that? I'm planning to use clustering, so unsupervised machine learning, and put all of my, so I still have to decide how many clusters I'm having, but at least I'm not saying that plant is that and that plant is that. And so I'm hoping that with using clustering on all of my, just the quantitative parameters, not the images at first, I will get several clusters of plants that are grouped together. Because, I don't know, does everyone know what clustering is? You're fine, right? Yeah, okay. And hopefully later, I would like also to try, I don't know, that's very experimental, I don't know if that will work, but I also want to do clustering from image, from image embedding, but that's the same, and see if my clusters relate together. If they do, it's really cool. If they do not, I still can use the supervised learning, the CNN thing, the transfer learning on CNNs. So that's it for now. Do you have questions on that?
Hi, can I ask something regarding the clustering part?
Wait, I'm putting you a higher volume. Yes, can you talk?
Yes, can you hear me now?
I hear you better, thank you.
So regarding the clustering part, would you consider maybe some of these methods that exist in cluster analysis for estimating the number of clusters? Do you think that this could potentially provide some more insight, or do you want the number of clusters to be fixed ideally in advance?
Oh, ideally, I wouldn't fix them in advance, but I didn't know it was possible. So wait, I'm gonna take my notebook and a pen. I'm still learning. So can you give me names of techniques that can do it, please?
There's multiple ones. As far as I know, the average silhouette width is the most common one.
How do you write it?
I can maybe write it in the chat.
Thank you.
If that's easier.
Andreas will give it to me. Okay, that's even better actually. I didn't know you could do that, but that's exactly what I'm looking for. Thank you. Do you know if it works well with data that is quite continuous?
What do you mean by quite continuous?
Yeah, I mean that I have, let's say drought is the soil moisture. I have anything from 0% to 100%. So I don't have like big classes that are super well defined, you know?
Yes, well, it really depends actually because of the definition of the criterion that is used to estimate the number of clusters. So it really depends on your data, but I don't see why it wouldn't work either. So,
cool.
May I ask, why do you need the clusters actually? Because, like you said, it's just like very living plant to a very dead plant, so it's like continuous flow.
Yes.
So, why did you need the clusters?
You mean instead of linear regression?
Yeah, sort of.
Because I don't have a drought index. I have plenty of parameters, and I don't know how to relate. I don't have a final score.
Right.
And I don't know how to create that score.
So then you're using cluster as kind of like a proxy.
Yes, exactly.
Or clustering. as a proxy.
Yeah, like to create that score.
But what you would actually want is the continuous number.
Yes, that would be even better. But I could ask Bayesian experts to help me, but I doubt it would work because we don't have drought doesn't exist. It's a concept we invented. Plants see more or less water, they see and more or less temperature. But yeah. Okay, is there anything else? Okay, so now what's next? So that's for you to decide. I have a few ideas that you could use for your posters. So there's a webinar. It's on Friday. It's during Crema's talk. So you cannot attend it if you go to her talk, but it's usually recorded. And this webinar is on artificial intelligence and machine learning in plant science. So it's supposed to be super cool. There are some people who are machine learning experts, some people who are plant experts. They're going to be trying to talk together, and I think some people who do both. So it should be interesting. In general, if you're interested in plant science, I recommend any seminar by the plant I represent. It's the American Society of Plant Biologists, and they make very, very interesting seminar. Can I send them the slides afterwards so that you don't have to write the... Yeah, okay. I would be happy to send you the slides if you're interested, because you will want, so that you don't have to write the whole web page. Okay, another additional material that I had in mind was a super cool literature paper. I don't know if in other fields you have those literature papers. We call them review in biology, which is super confusing because when you're reviewing a paper, it creates a lot of counterintuitive things. It's called Digital Agriculture for Small-Scale Producers, Challenges and Opportunities. So it focuses on subsistence farms, which are very small farms where people usually produce just the things they will consume, they will eat, which is a lot of farming in the world, actually, especially in low and middle income countries. And so, for example, here, that's a figure from the paper. It tells you, I really like it how they call it the through of delusionment. So basically they say how the hype for AI increases when you have more and more technologies, and then at some point you realize that it's not such a good idea, and then slope of enlightenment, I love it. And then at the end you realize that there are some stuff that can be useful, like call centers. Oh, I didn't talk about call centers. It's basically ChatGPT, but with a call, because you don't have internet somewhere in your field, so maybe. So you call someone, you say, my cow is weird, and they will tell you, they will do the chat thing and help you. Okay. And another thing that I had in mind was looking at the tools that I showed earlier that biologists can use to do AI, and think, if I was a biologist, what would I do with it? Would I be able to use it, depending on your different levels of, on your background. They have models, they have different tasks possible, you can download the training and the test data sets. You can have directly the notebook in Google Colab. Yeah, you can have fun with them. And maybe figure out, I don't know, a biological question, and if you were a biologist, could you answer that? Okay, so thank you for your attention.